All right, today we're going to crack a scientific case. We're going to follow a team of researchers on the hunt for this tiny but really destructive suspect, one that's threatening some of nature's most important workers, the honeybee. And this is the invader at the very heart of our case. Now, you might look at it and think it's small, but believe me, it's putting all of North America's honeybee colonies, our most essential pollinators, right, in some serious jeopardy. So that leads us to the big question, right? Can this dedicated team of scientists actually gather the clues, figure out their target, and come up with a plan to stop this pest before it's too late? Okay, so our story really kicks off with our main suspect making its grand and, let me tell you, very unwelcome entrance. So let's meet the villain of our piece, the small hive beetle. And look, this is not just some local problem. After it was first found in the US back in '98, this thing went global, like fast. It popped up in Australia, then started spreading all across Canada. We're talking Manitoba, then Quebec, and right into southern Ontario. This pest is persistent, it's successful, and it is absolutely on the move. You know, what's so wild about this is the difference a continent makes. Back in its native Africa, the small hive beetle, or SHB for short, it's just a minor nuisance. The local honeybees over there have actually evolved defenses to keep it in check. But here in North America, our European honeybees are completely defenseless, and that turns this little pest into a major threat. And the damage they cause, it is just devastating. The adult beetles and their larvae, they don't just eat the colony's most vital resources, the honey, the pollen, even the baby bees. No, they also, they defecate all over the hive. And their waste has this yeast in it that causes the honey to ferment and spoil, totally ruining the colony's entire food source. It's a mess. All right, so the threat is clear. Our scientific detectives started their investigation. The very first step, they had to track this intruder. They needed to learn its habits, its life cycle, basically find its weaknesses. And the playbook they're using for this investigation is a strategy called Integrated Pest Management, or IPM. You can think of it like this comprehensive approach that uses a bunch of different techniques, not just to solve a pest problem, but to prevent it from happening in the first place. The whole goal is to keep the bee colonies healthy and strong. So this whole IPM system, it breaks down into 4 key steps. First up, awareness. You got to understand your enemy's biology. Second, monitoring. You track their numbers to see how bad the problem is. Third, you set a threshold, which is basically deciding, okay, now it's time to act. And finally, you implement a solution. It's pretty much the scientific method, just applied directly to getting rid of pests. So using these specially designed traps, the researchers got their first big break back in 2011. What they saw was this huge spike in beetle larvae leaving the hives in May and June. This was the first wave, a coordinated exit, all heading out to go pupate, mature in the soil. And then clue number 2 dropped. Right after that wave of larvae left, the traps suddenly started catching way more adult beetles in the middle of the summer. What did that mean? Well, it meant a whole new generation had successfully grown up and was now coming back to reinfest the hives. And here it is, the final piece of the puzzle that really cracked the case of their life cycle. In September, the data showed a second massive wave of larvae leaving the hives, and that confirmed it. The beetles were successfully reproducing within a single season and creating multiple generations. The problem was compounding. Okay, so with the beetles' seasonal movements all mapped out, the scientists were finally ready to go from just watching to taking action. It was time to set the trap. And their plan was actually pretty elegant. Based on all that data, they figured they would target that first wave of larvae. But here's the clever part. Not in the hive, but right after they left, during their most vulnerable stage, while they were pupating down in the soil. The goal was to stop them from ever becoming adults, breaking that cycle and preventing that big second wave from ever happening. So what was their weapon of choice? It was a product called Permanone R. It's a broad-spectrum insecticide, which basically means it's designed to kill a whole lot of different insects. The plan was to use it as a ground drench all around the hives, creating this kind of chemical barrier that the pupating larvae just couldn't survive. And this brings us to the climax of the experiment. The clues were all there, the theory seemed solid, the plan was set. It was time for the takedown. So, the million-dollar question, right? After all that careful research, all that planning, did the ambush actually work? Well, the 2012 results started rolling in and they were, let's just say, surprising. In one of the apiaries they treated with Permanone, there was this huge spike in both adult beetles and larvae later in the season. That was not supposed to happen. And here's the real kicker. The data from an untreated apiary, the control group, it looked almost exactly the same. The beetle population exploded there too. When you put them side by side, it was clear the treatment had no real effect at all. So after all that brilliant detective work, the solution that seemed so promising, it just didn't work. Plain and simple. The plan had failed. But in science, failure is never really failure. It's just more data. It's another clue that points you in a new direction. So what did this mean for our researchers? It meant the hunt was back on. So, why didn't it work? The researchers had a few ideas. Maybe the dose that's recommended on the product label was just too low to be effective. Or, hey, maybe the chemical didn't soak deep enough into the soil to even reach the pupae. Every failure like this just helps you design a better experiment next time. So, where do our detectives go from here? Well, they're already chasing down other leads. There's another chemical treatment out there, but the big risk is that it can contaminate the honey. More promising, maybe, are things like special insecticidal traps. An Australian study showed those worked really well. Or even natural stuff like powdered limestone. And that brings us to the end of this chapter of the investigation. The first big attempt at a takedown failed, but in a way, it was a success because it ruled out a key suspect in the search for a solution. And it leaves us with this critical unanswered question. With one method off the table, what new science, what new trap will finally protect the bees? The case, for now, remains open.